Welcome to Ask the Expert. Today we have with us Dr. Adolfo Garcia-Ocana. He's professor and chair at City of Hope. He has a long-standing interest in growth factors, hormones, and intercellular signaling factors involved in pancreatic beta cell growth, survival, and function. His lab has shown that hepatocyte growth factor increases beta cell proliferation, mass, and function in vivo and improves islet transplantation by reducing the number of islets required for successful blood glucose normalization. They've also uncovered protein kinase C as a novel key kinase involved in growth factor and nutrient-mediated beta cell replication and function. And this factor controls the activation of mTOR, which blocks the activity of phosphatase PP2A, leading to increased stability and accumulation of the transcription factor MYC. We also have with us Jiaming Liu, MD, also known as Brian Liu. Most recently, and thanks to a pilot and feasibility study from the Einstein Diabetes Research Center, he's been studying the therapeutic potential of regenerative factors such as harmine plus extendin-4 together with immune modulation by temporary administration of anti-CD3 on remission of T1D in NOD mice. And they have filed a patent on these results. He's also implemented single-cell RNA-seq and snRNA-seq in human islets and human islet grafts in the lab, and we've recently published these results in a preprint in bioRxiv 2022. And I think they're going to be extending on some of that work here. And then finally, Randy Bayol Kang, a senior research associate at City of Hope Beckman Research, meticulous and analytical researcher with 2.5 years of hands-on experience with computational biology in the lab. A very adaptive team player with in-depth knowledge of data collection, mathematical analysis, and model building using machine learning for biological research. Thank you very much to this very talented team for joining us from the City of Hope in just outside of Los Angeles in California. And the title of this talk is Single Nucleus RNA Sequencing Analysis Reveals Novel Insights in the Human Islet Transcriptome. These data were just presented at ADA, and we're very excited to have you walk through this paper. Thanks so much for joining us.
Thank you, Monica. Thank you very much, and thank you to the Sugar Science platform for inviting us to give this talk. Good morning to everybody from the West Coast. I'm Adolfo Garcia-Ocana, as Monica just said, and a professor at the Department of Molecular and Cellular Endocrinology at City of Hope in Duarte, California. And with me are my colleagues in the lab, Randy and Brian Liu and Randy Kang. So we're going to talk and introduce our recent study that was published in Genome Medicine. The title is Single Nucleus RNA Sequencing Analysis Reveals Novel Insight in the Human Transcriptome. I'm going to do the introduction, and then from there, Brian will talk about a little bit about the methods, and then Randy will provide all the information behind the results, and Brian will finish with the summary and conclusion.
Fantastic.
Let me start with the introduction, just saying something very obvious probably to this audience, that is the diabetes results from pancreatic beta cell loss in both type 1 and type 2 diabetes. In type 1 diabetes, there is a dysfunctional beta cell at the beginning, together with autoimmunity that leads to a decline in beta cell mass, beta cell function, and diabetes emergence. And in type 2 diabetes, what happens is that there is an increase in beta cell function due to a need for insulin secretion, and is mostly genetically based as well as environmental based and lifestyle based disease that leads to exhaustion of the beta cells and diabetes. Have been postulated that pancreatic beta cell regeneration and replacement could be part of diabetes treatment, and there are many groups working on how turn different cell types of beta cells from hepatocytes, from pancreatic cells, from enterocytes in the intestine, or even, as we have been working, and that Monica introduced me, actually have been working a lot about beta cell regeneration, how to get more beta cells. There are many ways to do this, as I'm mentioning here, but one thing that was missing is perhaps how to analyze, in particular, human beta cell regeneration in vivo. Right? So this is a challenge that actually we pose in the lab. So when one day, after doing all these transplants, we decided, okay, let's study this cells at the transcriptome level, how can we do that? So we thought, okay, we have experience on human transplant. We could put transplants into immunodeficient mice, but then what? What do we do to analyze these cells individually? So we went to what had been published since 2016, and this is just a list of publications actually in that year, actually when everything started. Let's put it this way, on the single-cell RNA sequencing in human animals, actually, in which these studies actually designed or discovered the heterogeneity of cells inside the islets, and even inside of populations, islet cell populations, also the heterogeneity of those islets. So we thought, okay, if we are able to transplant human islets into mice, deficient mice, and then we can perform any treatment of those mice or any physiological challenge or any pathological challenge. You can think about hyperdiet, you can think about inflammation, you can think about hyperglycemia, you can think about anything you want, just to see what is the impact actually on those human islet cells. And after that, we could do analysis on, for example, you transplant islets in one kidney, you can take one kidney and do 3D analysis of the different islet cell populations inside of the kidney. But you can also take the other kidney with human islets and perform, for example, single-cell RNA-seq analysis. We thought, okay, we could have both analysis of the cells at the single-cell level from the single-cell RNA-seq analysis and transcriptomics, and also the immunofluorescence analysis and analysis of beta cell mass in that situation. So we went back and analyzed what it was done by then, and actually we found this is a great paper actually by the David Harlan group, Rita Bortel, and Agatha Jurczyk at UMass, actually, in which they were the first ones to do single-cell RNA-seq in human islet transplanted into mice. So what they did basically is to transplant 4,000 islet equivalents under the kidney capsule in an immunodeficient mouse, and doing multiple studies in the human islet graft. One of them was to do single-cell RNA-seq. Here you have a view of the islets that they transplanted and how the islet graft looks in the kidney, and how they got these 4,000 islet equivalents transplanted into the kidney, and how they did single-cell analysis and actually the identification of the different human islet cell types in that graph. It was, as I said, this is a pioneer study, but the point here was that actually they transplanted 4,000 islet equivalents, and the amount of cells that they recovered at the end, it was limited. So it was around 700 cells. So this tells us actually that probably just getting all the cells from the graft is not an easy task by enzymatic dissociation. So we thought, okay, perhaps we could implement a different method that would allow us to get more cells from the human grafts. In 2021, a pioneer study by Giorgio Basile in Rohit Karni group, also in genome medicine actually, they published the first single nucleus RNA sequencing in human allografts. And what they did actually is a very clever study in which they first did analysis of single cell and single nuclear RNA-seq from human in vitro, and found that UMAPs and the different clusters they clearly overlap in when they use the exonic reads actually for the analysis, and the number of genes that they found in both analysis, single nucleotide and single cell RNA-seq from the same islet cell population, it was very similar. They went to in vivo and applied this reference that they built with single cell single nucleotide into the human graph from the mice, and they found the obvious human islet cell types that they were expected from the human graph. So this was a pioneer study, as I said, and it was the first time in which single nuclear RNA-seq was used for detecting different human islet cell populations from human islet graphs. So basically, they use single cell RNA-seq and single nuclear RNA-seq, and they apply that to fresh and also frozen, that's important, culture or transplanted human islet samples, but they use exon reads only. And they found that there is a complete concordance in identifying cell types and gene proportions. Also, there was a strong association of global and islet cell type gene signatures. So after reading this paper, actually, we came up with several questions actually that in the lab that we try to answer with the paper that we have published. That is, if we use single nucleus RNA-seq and the nucleus contains mostly pre-mRNA with introns, what kind of information we could gather if we use exon and intron reads in our analysis. Can we generate an accurate reference annotation library with coding and non-coding region-based reads for human islets with single nuclear RNA-seq? If we include intronic reads, will there be any concordance in cell type identification and gene sets? I just mentioned that in the Dr. Kulkarni's paper, there was complete concordance using just exon reads. And the last question was, can single nuclear RNA-seq accurately annotate not only human cell types in vivo and their transcriptomic profile, but their heterogeneity? That was something missing in Dr. Kulkarni's paper, actually, in which the cells were studied, but there was no deeper analysis in terms of whether they could detect heterogeneity in a specific islet cell population, and what would be their corresponding biological process by using the gene set enrichment analysis. So I'm going to let now Brian to introduce a little bit the methods, and then Randy to go with the results of his paper.
Fantastic. Thank you very much for that really comprehensive introduction.
Hi, I'm just briefly introduce the bench work a little bit. Then the first, we just collect the human islets from the donor, and we make them into the single cell suspension. And for the single cell RNA-seq, we just go through the live cell selection, then move on to the check the cell concentration. For the single nuclear RNA-seq, we took advantage of the kit from the inventor and make it into the single cell, single nuclear suspension. Check the morphology with the microscope, and also we check the single nuclear concentration with the Contest 3. Then we just took advantage of the 10-genomic platform with the region kit V3.1, and to generate the cDNA and the library. Then send up to, for the sequence in the core of the wheel canal medicine. And then we got all of the FASTQ data. Then just pass to, I'm going to pass all of the analysis part to the Randy Kang. She's going to introduce how we got our result.
Okay, finally. So I will talk more about more algorithmic part of the business. So let me get the laser pointer. So practically, we projected both single cell and single nuclear data to the external reference, which is developed by the New York Genomics Center, and identify the cell type in relatively unbiased way, because we don't mark them and then we don't just annotate them on our own. It's more like a projection.
And Randy, is that resource readily available to all?
Yes.
Okay, great.
Yes, it's readily available by the coding and also by the app, so anybody can go in there and then project your data there. It's pretty simple and easy way to do that. But the thing is, since our data was large, so practically we had to download. I mean, we had to do it in more like coding way rather than app way. Then we perform the then we perform the differential expression analysis between single nuclear cluster only, and then define the single nuclear specific markers. So by doing this, and on the other hand, we prepare the separated data set, which is like a single cell and single nuclear integrated one, and for subpopulation study. So before we begin the actual analysis, we try to assess the similarity and differences between single cell and single nuclear data. So here, like the violin plot shows the differences between single cell and single nuclear RNA-seq. And then we use the exon-only library for single nuclear data. And this one, it's actually, we used an intron-inclusive data. So by adding intron-inclusive data, actually, we could increase the number of the gene per cell and also UMIs. So we try to create, better integration anchors for supervised clustering, which is like, projection, which will come after this slide. So as you can see here, around 71% of the genes are common between single cell and single nuclear RNA sequencing. And these genes will act as an integration anchor. And we integrate the single cell and single nuclear like here. So single cell is a very simple story. We have a reference which has a exon only single cell RNA sequencing data. So our single cell RNA sequencing data was projected near perfect score to the azimuth reference. But when it comes to the single nuclear data, it's a little different. So we actually had a choice whether we will use exon only database, referenced single nuclear data or intron inclusive references. And you can see like we actually had a significantly higher and improved projection score when we use the intron inclusive reference. So we decided to use it for the further analysis. Then you might ask that why can't you just use the common markers for the cell type identification? So upper part shows here, like actually it works well, but remember this is a single cell only data. It worked very good, and then you can see very clearly localized gene expression pattern for each given cell cluster. But when it actually comes to the single nuclear data, it's actually a completely different kind of story. So like the common markers don't actually work as clearly as single cell RNA sequencing data. And when you look at the expression pattern in UMAP, it gets even more hazy. So final solution was that we separated the single nuclear data and then performed the differential expression analysis against each cluster to all the other cluster. And then we compiled the 4 candidate gene markers. So alpha cell with a PTPRT, FAP, PTK4, LOXL4, beta cell with a zinc finger 305D, TRPM3, LRFN4, and Pluto complex, delta with a LRNFN5, AD8RB4, ERBB4, and KCNT2. So unfortunately, PP cell type, we found the 4 qualifying genes statistically, but it was not very clear as alpha or beta or delta cell. And then now we have a more localized expression pattern of the marker genes. And further, we confirm the expression of the one of our beta cell single nuclear marker, which is zinc finger 305D. And they were labeled in red. And then insulin secreting cell, which appears in green. So you can actually see the localization of the zinc finger 305D in insulin secreting cells by the in situ hybridization. Then we move on to the second phase of this study, so which investigate the beta cell subpopulation. So we integrated single cell and single nuclear data, and then cluster them with an unsupervised clustering algorithm. Then we name the cluster according to the canonical gene markers, like such as the GCG or INS, and using single nuclear genes such as PTPRT and zinc finger 305D. Then we separated the beta cell cluster and then increased the Lubin resolution to 0.8, tried to get some subclusters with unbiased manner with meaningful transcriptome differences. Then we actually named them as a beta 1, beta 2, beta 3 for convenience. And when you look at our favorite genes, aka insulin, we can see the stark differences between the single cell and single nuclear RNA sequencing data. So while beta 1 has a highest insulin expression, or like a near highest, very high insulin expression in the single cell data, but the thing is, when you actually look at the single nuclear data, beta 1 has a very low insulin expression. In the meanwhile, beta 3 has a high insulin expression pattern in the single nuclear data. But the thing is, in the when you look at the single cell data, you have a relatively lower insulin expression. Yeah. So I believe it's because the beta 3 cluster it's actually actively insulin transcribing. And then while the more beta 3 is, beta 3 is actively insulin transcribing cluster, and the beta 1 is more of insulin are actually exist in the cytoplasm, so ready to be secreted. So that's probably why we don't have much insulin mRNA expression in single nuclear data in beta 1 cluster. So when you look at the gene set enrichment analysis, we see the beta 3 is more ECM or interaction related pathways were enriched, and the beta 2 for vesiculation, and then beta 1 for the secretion-related pathways. So then we move on to our main interest and our specialty, the human islet grafts. So as two my bosses discussed prior slides, the graft should be processed in the single nuclear RNA sequencing data inevitably due to their highly fibrous character. So we transplanted 8 human islet grafts from 8 donors to 8 mice, which is like RET1 knockout, for 3 months is implantation. Then we harvested them and processed them in the single nuclear data. Then we harvested them into the process in the single nuclear data. We double confirmed that expression of the newly found genes on agemus and single cell and nuclear RNA sequencing integrate data. And also we double confirmed that expression of the newly found the markers on agemus and also external data as well. And the canonical, as you can see, the canonical markers doesn't provide a very nice expression pattern when it comes to the cell type identification. But the thing is, actually, when you look at this newly found markers, it shows pretty clear pattern and also very nice localization inside of the graft in vivo single nuclear data. So we projected our the human islet graft in vivo data to subclustered beta cell references, which is like the single cell and single nuclear integrate data. So practically, this is our internal data. So compared to the single nuclear in vitro data, graft shows a higher expression of the insulin in beta 1 cluster, and also showed significantly higher proportion in the beta 1 cluster as well. So further in the pathway analysis, beta 3 still shows pretty high and selective enrichment of the ECM and interaction related pathways. The difference between 2 and 1 was kind of hazy. So practically in general, the beta 1 cluster shows like high enrichment for the vesicle related pathways, and also the insulin secretion and processing metabolism pathways. So it more work as a continuum from the beta 3 to beta 1. And I think we believe it's due to the microenvironment and ongoing nutrition supplies, or even increase the mouse vesiculation, cause the differences between in vitro and graft in vivo samples, I believe. And again, this is new information, and the new markers, new marker genes, we have to kind of validate the expression in other people's external data. So at the moment, of this study, there were only 2 available single nuclear islet data. So we tested them both. So for Dr. Vasilis' data, we annotated a cell, as you can see here. We annotated a cell according to the agemus with the agemus projection. Then we confirmed the localized expression pattern of our new marker genes, especially endocrine cells. And then it actually shows pretty decent localization of the gene expression in each given cell cluster. And also, we collect the data from the Dr. Luca Tosti. And the Dr. Tosti's data, it's only available through the web application in the United States, because it was very hard to access their data through the, so we use their,
Randy, can you repeat where it was, accessible from? Again, I couldn't hear you.
So Dr. Tosti's data, it's available through the app, online application. So you actually don't have to download their whole, the vascular files and analyze on your own. You can just simply go into their websites and then just type your gene, and then you can see the expression there.
Got it.
So, so we tested out the 3 genes, the PTPRT4 alpha. As you can see, it has a very nice localized pattern in their data set as well. And the zinc figure 3F5D in beta cell, and the LRFN5 in delta cell. So by the way, the Dr. Tosti's data is a pancreatic tissue data, so rather than the islet only. So I think these new marker genes, for the single nuclear sequencing, actually can work in the tissue data as well. And then we somehow should verify the beta cell subclustering data as well, but it was very hard to find it at that moment. So here we get the list of the expression gene from Dr. Dorell's paper. He brilliantly divided the beta cells into 4 groups according to the CD9, and ST8SIA1 expression. So like, I mean, this is kind of a complicated. So like, long story short, practically CD9 positive cluster is insulin responsive, according to the Dr. Doral's publication. And the negative is definitely less responsive. So we also observed the CD9 positive beta-3 cluster has a localized insulin expression in our single nuclear data only, while beta-1 cluster has high insulin content in single cell. And also this cluster shows a high GIST enrichment for the processing and secretion pathway in GC data. So practically it kind of, go along with our data, with the Dr. Doral's data. And also further, like, gene co-expression pattern, as you can see here, showed a similar trend to the Dr. Doral's data as well, both like, single cell and then single nuclear data. And then beta-2 shows some kind of, something in between. So I think we verified our finding with previous publication relatively well. And then I'm gonna pass this the whole presentation to Dr. Liu for summary.
So to summary our study, first we found the single nuclear RNA-seq can actually annotate the human islet types in vivo, transcriptome, and heterogeneity, and their corresponding biology process. And also to include the intron pre-messenger RNAs in the single nuclear RNA-seq analysis provide great accuracy and superior correlation score than using only exon reads for the human islet cell type annotation. And our study is the first reference annotation library, including non-coding and all coding region for the human islet single nuclear RNA-seq. In our study, the top 4 differential and the selective expressed gene in the human endocrine cells derived from the single nuclear RNA-seq are not canonical gene, which is used to define the single cell population in the established the gene set. And to integrate the single cell RNA-seq and the single nuclear RNA-seq data set in the same human islets can detect the 3 different beta cell subpopulation with the different transcriptome profiles and potential different fundamental biology process. And also our study shows the same global islet population and the 3 different beta cell subcluster appeared in the human islet graph as well. And then the last, the proportion of the cells in the 3 beta cell cluster in vivo is changed as compared to the in vitro study in the manner that favor a mature and fully functioned beta cell phenotype over an incomplete differentiated beta cell phenotype. So to conclude, these observations provide a new lens through which to understand the human islet cell biology, and also provide the new tools to explore the human islet cells biology in vivo. And the last, we appreciate all of the lab members from my PI, Dr. Adolfo Garcia-Ocana's lab. And also we appreciate all of the collaborators from Mount Sinai, City of Hope, Dan Faber, and UC Davis. And we appreciate all of our funding agency. And the last, but importantly, we appreciate all of the islet donors. So now we are happy to answer the question and have a discussion with you, Monica. Thank you.
Wow. Brian and Randy, in the entire Adolfo Garcia-Ocana lab, that is amazing work. And congratulations, very meticulous, very detailed. It's an innovative approach. And we commend you that you went back into some of these published data sets using new tools to pull out some, really novel insights. And I mean, I guess before we have a lot of people attending this, so I'd like to open it up to questions. Before we do, I'd like to ask you, this whole idea of the beta cell subsets, which you've identified, how might you hypothesize or think about it in context of the patchy attack on the islet cells that happens in type 1 diabetes?
Sorry, that's an excellent point. I mean, the reason why we start all this, as I said in my introduction, is to analyze human islet cells in vivo, I mean, through islet transplants, and then in different situations such as inflammation or high fat feeding, etc. But as Randy said during his presentation, we can go also to human pancreas data sets right now that they are, for example, in HPAP here, and analyze, for example, these different subtypes, actually, like beta 1, beta 2, beta 3, in samples from type 1 diabetics or type 2 diabetics, and see whether actually there is a variation in the numbers or in the transcriptome profile of these different subtypes of beta cells, and see whether there is one that is more favor or less favor, depending on type 1, type 2 diabetes, and analyze those cells in particular. So this is just a platform actually that these two guys here created actually for everybody to go and now do this type of analysis on data sets that are already available, or newer studies in which you transplant islets or in vivo pancreas and you perform this analysis as well. So it's kind of an initiation on studies on single nuclear ISIC and beta cell heterogeneity.
Can you do me a favor and not share your screen anymore so we can see your full view? And then I'm going to open it up to questions. You can drop a question in the chat.
We're always happy to answer any question also by email. If they send anything by email, any question, happy to share all these data sets as well. We put all these data sets in the manuscript and repositories, so it's easily available to everybody. Happy to share all this knowledge.
Okay, if people are a bit shy, I guess, or maybe just stunned by the enormity of this excellent data. What are your next steps in the laboratory?
So as I said before, I mean, we can talk about this more, but is data mining. So now, for example, at the ADA, Randy presented alpha cell subpopulations as well, and see how they can project into a different cell type, right? I mean, how they can, using very advanced algorithms. I'm not going to talk about that because I'm not an expert on that. I'm biology, so I get lost on algorithms when you start talking about that. But using all the latest tools, actually, how to analyze the trajectories of the different cells going from one stage to another, right? And he presented this data, for example, for alpha cells, and how there are different subtypes of alpha cells and how they can project into beta cells, and how that changes between in vitro and in vivo. That's an important issue. So
Yeah, very important.
Cells change a lot whether you are doing your experiments in vitro or you are doing your experiments in vivo. And
Okay. Well, that's good. Yeah, go ahead, Brian. There's several questions I wanted to read out.
Yeah, and also I think the data set as the reference can be used for the future study, especially, for example, the T1D patient. They got the paraffin embedding the tissue blocks. We can just take part of the tissue and make them into the single nucleus. Then we can project on our data set. Then we can do the investigation how the T1D patient, like the pancreas or the islets, got any changes or the impact by the pathogenesis. So I think our data is really useful as the reference for the future study, especially for the T1D.
Oh yeah. No.
In terms of like computational usefulness, so generally, like, single nuclear like has a better proximity to ATAC sequencing as well. But I mean, people do the multi-omic recently anyway, but the thing is, if you don't have that in your hands, so that like actually if you have a single nuclear data for sure, and then you know the cell type very well, and then you can project the ATAC data there more, better like way, better integrations and then better UMAP coordinates and stuff. Yeah.
Yeah, no, that's an excellent point. Here are some questions. Hi, great presentation. Can you repeat the name of the platform or app that you mentioned?
So Like for the analysis, I use the SORAT, ScanPy, SCVALA for the RNA velocity. It's like kind of, well-built analysis R coding packages. So you can actually simply just download it, and then they have a tutorial too. And also, if you're actually interested in, like, you can come to my GitHub, which is written in our genome medicine paper. So I actually uploaded all the code that I used to analyze for all the data that I presented today. And also, like for the Dr. Lukatotis app, you can refer to his paper. Dr. Toast, I think it's Dr. Toastis lab. So if you go to their paper, they do have a link to the application. I don't remember the name of the app, but I think it's written by the Shiny app. But and also it's in my paper too, so you can just take a look and then just use it for your analysis. Yeah.
Fantastic! How much sharing you guys are doing, and how collaborative you're being. It's just amazing. Another question. Sorry if I missed this. In single nucleus, do you know the cells were viable before extraction?
So yeah, that's a good question. That's why in our protocol, we do use the live dead cell selection before we make any single cell on the single nuclear, the RNA-seq. So, and also actually we run the, when we modify our protocol, we do run the flow cytometry to check the cell death and even the viability of the cells. That's for sure. We know all of the cells, most of, like around the 95 or 98% of cells we process is the viable.
And also, like, to add Brian's points, the beauty of the single nuclear sequencing, like, cell doesn't have to be alive.
Yeah.
So
I mean, they can be frozen.
yeah, it can be frozen or it can be dead or perforated. As long as your nucleus is intact, you can actually extract the RNA-seq from inside of it.
Both good points. Any predictions on how the different beta cell subtypes may change during diabetes? We did talk a little bit about this, but
Yeah, I mean, that's an excellent question. Actually, that's exactly what we are doing right now. But I mean, yeah, well, we are mining the different data sets that are out there from type 1, type 2 diabetes, and see whether actually these different subtypes change in number or the transcriptomic profile, not just beta cells or alpha cells, for example. So yeah, now is the time to use this platform to advance in diabetes settings.
Yeah, and also we are currently doing the like a trajectory modeling for our next paper. So when you actually look at the T1D or T2D or like in a control patient, actually it moves all in different kind of direction, and then we're gonna like put it in the bioarchive like relatively short time. So please stay tuned.
We are very much tuned into this group. This is really amazing, and cannot wait to see that paper. Here's one more question. Sort of somebody tapped on the whether the cells are viable, and now someone's asking, what's the method you use to check the nuclei quality, as you did when you did snRNA sequencing?
So for the nuclear, first we have to check based on the morphology part. We have to use the lab field to check the overall all of the intact of the nuclear. For example, if the nuclear become the creepy, a little bit shrinked, which means that the method doesn't very good. And also we have to use the fluorescence staining. I just use the AOPI staining to check if they're mixed with the single cell, which means that I didn't process the nuclear thoroughly. That still mix some of the single cell. If there's the green fluorescence, and also that's pretty much we control. And also I think Randy put the really good algorithm when he analysis. For example, the mitochondria the proportion is really high. He's going to remove that part of the nuclear from our data set.
Yeah, like if you're more interested in the QC, like just send us an email, because there are like a deep neural network based algorithms which is available online, I mean, not online, available through the code, and like there is another ways, and also you can like even calibrate the MBR mRNAs by like a different type of algorithm. So actually, I wrote most of that in the paper. So like if you are okay with some technical terms, you can try to read it and then go to like a developer's website and you can try it out. Yeah.
Fantastic. Everyone says thank you so much. I would also just ask, this lab seems like it's kind of on fire with innovation. Are you looking for graduate students, postdocs to join you?
Oh, yeah.
Yeah. Over there at City of Hope.
Actually, we just moved to City of Hope 4 months ago. So we were at Mount Sinai before. The lab is growing. We have a new research technician in the lab, but we are looking for postdocs or even students who want to join our lab. Definitely, this is an opportunity right now. We are doing a lot of studies in terms of transcriptomics and in terms of many other different aspects of, for example, beta cell regeneration. So it's the time to join our lab right now.
Fantastic. All right. So if people have interest, reach out. There's a lot of exciting things going on at City of Hope over there. You've got the new directorship of Alberto Pulesi. John Cadiz heads up HERN there. We've got some very exciting shining stars in research there. And so great to hear about what you're doing and get you on the map, and so that everyone can sort of follow what you're doing and reach out for collaborative efforts. Thank you so much for sharing this, and congratulations. It's just a lot of work, but I mean, you really unearthed some really interesting data here. So thanks again.
Thank you for your invitation, Monica, and congratulations on the Sugar Science platform. It's fantastic, actually.
Thank you. Have a great rest of your day.
Thank you very
so much.
Bye-bye.
Bye.